Well, good afternoon, and I guess good morning to some. Thank you for joining the Foundation Fighting Blindness on our Proposers Day for the Translational Research Acceleration Program. My name is Chad Jackson. I'm the Director of the Preclinical Translational Research Program at the Foundation Fighting Blindness. Before we jump in, because this event is all about you, the researchers, and giving you the ability to ask us questions on the application process or what it's like to be a TRAP awardee. If you have a question, please use the toolbar within Zoom, within the Q&A section to put your question, and we will try to answer it as soon as possible during the presentation so that you have all the pertinent information that you need. This event is being recorded and will be available for viewing at a later date and time if you go to the Foundation Fighting Blindness's YouTube channel. With us today, I also would like to introduce Dr. Mahdi Farhan of the Usher3 Initiative. In a little bit, he will be giving you his remarks as a TRAP awardee, as he was part of the inaugural class of the FY21 application cycle. In today's meeting, I will be giving the overview here in a second. And again, Dr. Farhan will be giving you his remarks, and then we'll try to leave a good amount of time during the event for you to ask questions. And please come with any questions. We want you to be able to submit the best application possible, so let's not leave anything unsaid during this meeting. So here is just a little overview of the Translational Research Acceleration Program, which we will be terming TRAP from here on out. The reason that this program is so important is because we understand that people with inherited retinal degenerations need an increasing number of therapies to combat the complexity and uniqueness of these diseases. And as well as we need something to combat dry age-related macular degeneration. So the mission of this program is actually to accelerate the science going on at the bench toward the clinical use through an IND approval and to attract third-party funding for the advancement and development of new therapies. If you look here in the middle in the schematic here, we're really laying out there's three major research areas that we're looking to fund. And we kind of left them as broad and as big as possible to include all therapies that sort of are coming online that could potentially use as therapies. So we're looking at the one section called novel medical therapies. This includes things such as small molecules and biologics. And we'll go deeper into each one of these categories throughout the presentation. Also genetic technologies, such as things as AAV-based therapies, as well as non-viral constructs, as well as gene editing. And then we have regenerative medicine. So this would be the use of stem cells or cellular scaffolds, or the mix of the two, in order to restore damaged retina. What's unique about the TRAP program is that we use a proactive management structure. This means that awardees are actually a part of a team. So very much a team effort, which includes monthly updates in which the awardee will give in partnership with a project mentor that the foundation assigns the awardee, that they work together. Usually this mentor has industry experience and will help sort of shape the program in order to meet the needs in order for it to move along to the next stage. Also, each project is overseen by an advisory council, and these are individuals with a sort of swath of experience, again, in order to advance this technology. And then coming out of the accelerator, we really hope that this garners the attention of either new partnerships or third-party investment in order that the technology has a life after our funding has run out. And ultimately, what do we want to see this result in? We want to see this result in an investigational new drug filing, establishment of a clinical trial, and of course, a commercial product. So let's dig a little bit further into the novel medical therapies. This is really sort of using chemistry and biology as well as bioengineering to create some things like small molecules, the tried and true, biologics, optotherapies. This includes optogenetic constructs, or sort of using light in general in order to drive cellular responses, and then also alternative therapies. And what we mean by alternative therapies here is that of the three main categories I mentioned earlier, your technology may not fit squarely within any of those. However, we would like for, if that is the case, we would like for you to, if you submit, to put your technology under the novel medical therapies, as these are being directed to a particular subcommittee for review. So we've already let those reviewers know that any sort of alternative approach they will be reviewing. I will stop here just to say, before I get into genetic technologies, that if you have not done so yet, please go to our website and look for the TRAP webpage. It has all the necessary information when it comes to application and the things that we're looking for and the things that are covered here in document form. So you can go there, you can download the documents, and it'll give you all the information you need in order to apply for this award. Now, genetic technologies. When we say that, we mean it as the manipulation or modification of human retinal gene expression to alter biological properties of living cells and tissues with the goals of treating IRDs, or dry age-related macular degeneration. So this can come in a wide sort of sweep, whether it be DNA, RNA delivery, even protein, if your construct is actually producing a protein. And then they're also looking at delivery mechanisms, whether that be AAV based or viral based of some sorts, or even non-viral based, such as a lipid nanoparticle, or even any other sort of emerging technologies where you're able to deliver genetic material. So this would be coming in many different phases, such as antisense oligonucleotide research, or even gene silencing technology. So we are leaving these categories as broad as possible. So if you feel like your technology fits within the scope, please apply underneath this category. And lastly, as far as the main topic here, we have regenerative medicine. And this is really using stem cells and cell sort of platforms, or the combination of the two, in order to restore or rejuvenate the cellular process and cellular damage of the retina. And whether that's seen in an IRD or dry age-related macular degeneration. The one thing to pay attention to is that you must also solve any immunotoxicity issues associated with that. So that has to be a part of the research plan. We are looking forward to understanding more about this process as we see clinical trials already in this space, but we're excited about the future of this and looking to fund more research in this area. So if you even know someone who is looking to do a project in regenerative medicine, please encourage them to apply. I see we have 2 questions here, so maybe I'll stop for a second. Oh, we have one question here. It says, can I submit more than one TRAP LOI? If so, how? The submission link sounds can only be used for once. I say that you can submit more than one application, and we will speak to the people who manage our submission website. I would suggest you trying to submit the different application underneath a different email, if possible. So if you use a different email, you should be able to submit more than one application. However, I will say that the applications need to be dramatically different from one another, as we will not consider something along within the same realm of each other. Thank you for your question. Now we're on to the funding mechanisms. And please excuse me for having so many words on this slide and hard to read. But like I said before, you can go to our website and you can download all the documents necessary to apply. All this information will be covered here. So the 3 types of funding mechanisms that we have available are tried and true, as are the foundational grants. So this is what falls underneath the TRAP, and we'll go into the different TRAP criteria in a minute. And these support the preclinical research, in which is the main goal of the TRAP program. But some things you need to consider: that the project should address therapies that are genetically driven. So we understand this. One of our main focuses is inherited retinal disease. So make sure that there's a genetic driver when speaking about that category. Projects should advance in looking to accelerate an actual sort of in-hand technology. When we talk about preclinical development, you need to have at some level, some proof of concept. So meaning that you have a potential small molecule, potential biologic, and potential gene therapy that you can actually advance. We cannot just advance an idea. You actually have to show some type of data that shows that there is some type of product that can able to be advanced within this funding mechanism. Also, the research should be hypothesis driven. So that means that in your application, you need to succinctly state that hypothesis. However, there is another caveat. If your research is at a very advanced state and you're only looking for funding in order to do 1 or 2 critical experiments before you're looking to move into IND filing or to attract third-party funding, they may not be hypothesis driven at that point and will be considered for funding. Also, lastly, what's very useful is for us to be able to understand if you are successful, what are the next steps for your research? So somewhere in the application, please convey that. If all goes well, what are the logical next steps in order to advance this technology? And lastly, we want to make sure that the technology that you're trying to advance has intellectual property protection. So we want to see a plan for that. If you do not have that currently for your technology, we would like for you in the application to speak directly to that. It's very important in the development process that is the case, and will help you attract third-party funding moving forward. The other 2 categories that are less used in the funding mechanism are an Ffb Cost-Share Partnership. This is where we provide in-kind funding with somebody who's looking to use part of their funding but needs a little bit more in order to get their work done. And last is the Ffb Advisory Partnerships. These are for people who are looking, maybe has this technology and already has funding in order to advance their project, but are looking, but may not work within the retinal disease space. And maybe they're looking to partnership with the FFB in order to get access to our science advisory board, our advisory councils, and management and oversight to try to advance that particular technology. I will pause for a second as it looks like we may have another question. Here, the question is funding available to commercial entities or just academic investigators? We are keeping the TRAP program as flexible as possible. Therefore, we are making this available to companies as well as academic partners. There is a difference in our award terms, which I will briefly cover later. However, funding is available for all. The main goal of the TRAP program is to advance technologies so that the people who need them can get them in short order. So here, if we look, there are different categories that you're able to apply to in TRAP funding. Now, the first one here I have listed as a TRAP PPA. Now, this year we have released a TRAP PPA, which stands for Program Project Award. But this research is usually a directed call on a specific topic that usually comes out of like a deep dive workshop. This year it's on ocular inflammation caused by viral vector exposure. I will go over that in a minute, so we won't have to sort of labor on that. That award will be a $1.5 million award that's distributed over a 3-year period. The other 3 categories, which is sort of our bread and butter when it comes to TRAP funding, is our TRAP 1, TRAP 2, and TRAP 3. And these sort of designations sort of identify at what level your research currently stands within preclinical development. TRAP 1, of course, is earlier on. We're starting really from target validation to proof of concept. So if you're very early on, so you have something in hand and it looks like it has potential, you may have one experiment done that shows that it has some sort of efficacy in, let's say, stopping retinal degeneration, but you haven't done any others. You would be considered for TRAP 1. TRAP 2 is really through lead optimization, through drug candidate selection on up to IND enabling studies. And then TRAP 3, we leave this as our most flexible stage award. Let me go back to TRAP 2. It is a $1.5 million award distributed over a 3-year period for a total of $1.5 million in funding. And TRAP 1 is up to $300,000 per year over a 3-year period, up to $900,000 in funding. So lastly, we have our TRAP 3 award, which is a a one-time, up to $1 million one-time award to do late-stage projects that need one or more critical experiments in order to sort of get them over the transition hump. And that would be either in order to collect data for an FDA IND filing, attract venture capital investment, or the logical next step of the project. Now, as I mentioned, this year we have released a program project award for what's termed the viral vector ocular inflammation. And the reason being is last year, Dr. Brian Mansfield led a workshop on this particular topic of saying that when people or animals or people are giving viral vector-based ocular treatments, most often you will see inflammation associated with that level of treatment. And moving forward, that actually can affect the efficacy of that treatment. So moving forward, we would like to stand up this program in order to establish research that can help mitigate that situation. So the research priority areas for that particular PPA are ocular inflammation pathways during retinal degeneration, viral vector-induced ocular inflammation activation and progression, and ocular inflammation mitigation. In order to apply for these, some of the key criteria is that it must be led by a single, it must be led by a single project leader. Also, there needs to be and the reason that we're calling this a PPA is that usually these topics are too large for one lab to tackle. So we would like to leverage the expertise of at least 3 projects within this total PPA in order to mitigate some of these processes. The research needs to address one of the research priority areas that are listed over to the left. And you also must have a particular intellectual property strategy, how to protect the IP that comes out of the research. And I will take a pause to answer a question. The question is, does the funding have to be distributed over 3 years, or could it be over 1 or 2 years? Great question. I would say the simple answer is yes, it can be distributed over a short amount of time. However, you must justify that cost. Although the program is called the Translation Research Acceleration Program, we want to be sure that you're able to do the quality of work within that time frame. So we have sort of based it out over 3 years in order to sort of meet the expectations of the timelines we see fit. However, if you're able to adequately show that your execution plan requires only a 2-year basis, but the funding up to the total level, and you're able to meet the milestones and goals set forth in the application, and they review well, yes, we can definitely distribute the time in less time frame. Great question. So, as I mentioned before, here are some of the eligibility criteria. You must sort of adhere to one of the research priority areas again, and this is in the TRAP funding, not the PPA, but both of them you have to sort of adhere to their particular research priority area. And again, I will direct you back to the documents that are on the TRAP webpage. Download those and follow those directions. That will have all the information you need to apply. You will have to be able to clearly state how the proposed research will be geared toward developing a product to address an IRD or dry AMD. You also must use the assays that are appropriate and favorable to FDA for IND filing. So this is one of the, I think, the biggest sort of nerve-wracking issues, especially when maybe a natural animal model does not exist. But this is why we have industry professionals and an advisory council to help sort of guide you along that path. So I would say in the application process, make sure you outline how your research plan, the assays that you're going to use, is going to give you the best data possible in order to move this technology toward getting an IND filing and toward the clinic. Of course, you must adhere to one of the categories. We only have so much funding, therefore we're going to have to put you in one of the sort of trap boxes, and you'll be funded at that limit. There will be, we do not have funding for cost overruns, so when you're developing your budgets, please take that into account. Now we're talking about the applicant's criteria. Again, you must have a leadership position. You may, whatever organization or institution that you're at, you have to be able to manage the money, be able to conduct research with the support of the institution that you're at. You are able to use collaborators, core labs, or commercial CROs to execute any portion of the research that you see fit in order to meet your goals. Please just outline that in your application. You must be able also that the personnel that you're going to use has the skill set and to execute the work. And also that the location you're going to execute the work has the appropriate equipment and space. Yeah, that's pretty much it for the criteria. There's other nuances, but again, I'll direct you back to the documents on the webpage. Please print those out, review those. Now, just a little bit about the TRAP selection process. First, we'll start with the publication of the RFA, which we're already in. Right now, we're at number 2, the Proposers Day webinar, which you'll be able to ask questions. And then we'll have the review of the LOIs. The due date for those are July 22nd. After we've had a chance to review, you would then be notified whether or not you've been invited for a full application. And then you will submit a full application, which the full applications are due October 30th at 11:59 p.m. At that point, they will go through our review process. They will go through a scientific advisory board review. At times, we will ask external reviewers to review your application. However, all of our reviewers are under a confidentiality agreement. Therefore, we will protect whatever you submit. I want to sort of foot stomp that we take confidentiality very serious at the foundation. And if you ever have a question whether or not where your information or where your application is being shared, we'll be more than happy to provide you with our process. While who's reviewing your application is a blinded process to the applicant, but we will ensure that everybody who's looking at your information has signed a confidentiality statement. After that, there's selection and notification, then a negotiation of terms, award terms. I'll stop and mention that if you are interested in understanding our award terms deeper, please reach out to me for the simple fact that we do have particular terms in there when it comes to technology transition. Mainly, the foundation also has a venture capital arm called the RD Fund. If you would like, please go and search for the Retinal Degeneration Fund in their own website and review their process. But what we'd like to treat it as is a natural transition from the TRAP program to the RD Fund if there's commercial application for the technology and that the technology is ready for third-party investment. So the foundation sort of reserves the right to take a first look at that technology for funding. And the way we like for our applicants to think about it is that if you are successful, you already have a third-party interest in the foundation in helping you continue along with the development of your technology. After that, there's an award notice and kickoff meeting, and then you start your research. I'll take a pause there to answer a few questions. The question is, excuse me one second, I have to increase the size so I can see here. It says we have, excuse me. Okay, we have a minimally viable AAV-mediated gene therapy product for AMD GA treatment. We have an efficacy and safety data on 2 mouse models with ROA. We want to get large animal data to collaborate for. Do you support multiple animal models to minimize cost? Yes, we can. We will support as long as it fit pretty much the research, as long as it meets the guidelines that we set forth as far as any sort of IUCUT protocols or institutional review, things of that, it meets all the ethical issues, we will support multiple animal studies. However, all that must fit within the budget of the award. So like I said, we can't do any cost overruns for projects. I hope that answered the question. If not, we can dig more on that into the Q&A. Just sort of doing a little publicity for the program. These are our current awardees, Dr. Tom Ray of Washington University, Dr. Stephen Sang of Columbia University, Dr. Rob Collins from the Radboud University, the Netherlands, Dr. Madi Farhan, who is here with us today, Dr. Paul Yang, Dr. Hendrik Scholl. And one person I left off there, and that is my mistake, is also Dr. Barb Rohr of the Medical University of South Carolina, is also a TRAP awardee, and that is my mistake, and I apologize for that. She is also a TRAP awardee and was actually the first TRAP awardee awarded through an off-cycle program to looking at mitigating dry AMD. I'll pause for another question. The question is, if there are labs in different institutions collaborating, does this work similar to the NIH subcontracting / individual institution indirect cost stipulations? Apologies if I missed it, but are those stated budgets total budgets, including indirects? Yes, the budget that you submit needs to be the total budget for the entire project, including any sort of subcontracting. Also, we will only have an agreement with the PI and the primary PI and the PI's institution. Therefore, any payments that need to be paid out to subcontractors or collaborators will have to go through the awarded institution. And thank you for it. We also do not pay indirect cost, if that makes sense. So that is one caveat of funding from the Foundation Fighting Blindness, that we do not pay indirect cost. Another layer of review for the TRAP program is the TRAP Advisory Council. And again, this is a mixture of industry professionals, clinicians, engineers, intellectual property experts, and regulatory specialists that are really, just in general, looking at the projects on a quarterly basis and giving their opinion on how to best move the technology forward toward the clinic. And that's there, and that wraps everything up. So each of these bullet points I'm not going to go over. However, that is the main job of the Advisory Council. And here are who currently makes up the Advisory Council. You have Dr. Nima Mayhew, Janet Cheatham, excuse me, Jed Chatterson, Mark Fields, Ashwath J. Gopal, and Hema Ramkumar are all part of the Advisory Council. And all of these individuals have signed a confidentiality agreement and will keep all the information of your project confidential. So here, just to reiterate the goals and key dates, we're really looking forward to advanced projects toward IND filing, hopefully the establishment of a clinical trial, and then ultimately a commercial product that our constituents can use. Key dates are listed here to the right. Of course, today, Proposers Day. Remember, letter of intent due July 22nd. Full application invites will be, people will be notified, hopefully, I will say that, by April 15th. Application due date will be October 30th, and then the review applications will take place between December of this year and January of next. And our anticipated award date is March 22nd. With that, I will stop sharing my screen and turn the presentation over to Dr. Farhan. Stage is yours.
Can you hear me?
Yes, we can hear you.
Well, good afternoon, everybody. I think I'll start from the beginning. Almost a year ago, I was sitting, listening to the same webinar. And I think, the whole process is really summarized very well by Chad in the slide entitled TRAP Selection Process, which has a nice 9 steps. And the first thing we really did, me and my team at the initiative, is really to say, is our program aligned with the objective of the TRAP award? We are working on a small molecule for ASHR3, which has retinitis pigmentosa and deafness. And we had a candidate selection. We've done all the biological work. We've done the basic screening for safety, the hair, the mutagenicity, the 2 weeks toxicity study. And really we needed to do almost the final pre-experiment, GLP experiment, to be able to file for an IND. So that's really fitted very well with the idea of the acceleration. So now we are in a late preclinical, and we needed to do critical experiment to accelerate it so we can file an IND and hopefully start clinical trials. So when we saw really the fit between what we're trying to do and what the TRAP award mission is, we felt it's really the appropriate to go and apply. So I'll tell you our experience before we applied, during our application, and after our application. Before we applied, we attended this seminar, so we had a really good idea. Me and my colleague, Georgia, we actually went through and read every document on the net, and we looked at other program being filed. So we had an idea what to do, what basically to do. And through that process, we had a number of questions. And not because Chad is here, he has been absolute help for us. So whenever we had a question, we either had an NGTC or we shoot an email. And we usually really clarified a lot of our question, how we actually need to go through this. After we decided to submit, the first step was in the process to write the letter of intent. And I think this is very, very important, because basically what you write in the letter of intent will allow people to make a judgment whether you need to submit a full application, i.e. your goals, your projects, your timelines are really fit within the TRAP philosophy and the TRAP mechanism. The few lessons I have learned is really that in letter should clarify what is your ultimate goal, where are you now, what's your road to the next step, and how you're going to do it, and most important, how long it's going to take you and how much it's going to cost. So these are all the key. Our letter was 4 pages, but it covered all this essential. We submitted in the letter, and then we had about 19 weeks later that we can submit a full application. We submitted a full application, which is really expansion on what we have done in the letter, except there was a few things this is the first time we submit, did not really realize we need to do more homework and write. And this one is really what Chad talked about. We all knew about the science background, the experiment we want to do. One is the patent issue, the technology. Is it protected? What's your plan with it? That we did not pay enough attention, so we had to write in that. 2, a detailed chart of the program, a detailed Gantt chart, and where the milestones are, and where is the risks are, and what's our alternative. 3 is the collaboration. There is part of the application. It talks about collaboration, whom you're collaborating with, what's the purpose, what's the relationship. So those were not really covered in details in the letter, and we had to spend some time on them. But overall, we find the actual application is really straightforward. It's not very large, and I say large in comparison with the previous application we did for the NIH. The instruction is very clear and is always held at hand. You can actually always ring and ask, I'm having trouble with the budget. Do I include this cost or do I exclude this cost? Is 10% sounds reasonable or doesn't sound reasonable? So all these questions were really answered by the TRAP and by Chad particularly, almost on a daily basis. So we had all the support we wanted, and then we waited for the application. It went through the process, and then luckily we had a contact both by emails and by a phone call from Chad to say, we have won the award. And that was very exciting, because the Ashara 3 initiative would not have ever really could find the money to progress the program in order to do the funding. And the reason is because the initiative actually was founded by a patient and her father. And it's not a big company, and it's not a large university institution. So it's really a small charity trying to find a cure for the Ashara 3. And this is fundamentally important, because throughout the application, we have to say, why is this important if successful? What would it do? How would it change people's lives? What's our plan to take it forward? And what Chad said at the end, this is really important if you are in the late stage of preclinical development. Looking at the end of it for IND filing, looking for a partner to help you out, looking for a partnership within the FFP, that's all important consideration. So the first thing we had to do is really look at the grant award and look at the terms and the conditions, discuss those. And again, we should have come back within 30 days of our final signature. But because we had to consult with our advisory board, our scientific board, actually the FFP has been very, very generous with us. And they were very flexible, and we extended the timeline in order to get a good discussion. And eventually we signed it. And the first thing we had to do is really have a kickoff meeting. That kickoff meeting, basically, we have to go through again the Gantt chart, again the budget, again everything else, and be on the same. And I must say, from that first kickoff meeting, it stopped being the funder. It's actually become part of the team. I feel already I'm part of the FFP, because it's clearly once they believe in your program, they're willing to fund it, they want you to succeed, and they will help you in every way they can. And it's not only we have a mentor who is an experienced person in industry to help us out, we also had access, whatever question we have, to the whole clinical scientific board of the FFB behind the scene. We also have whatever data the FFB have and they can share with us. So really we had much help beyond just the funding and the advice. And we have a monthly update meeting where we basically say, according to the timeline and according to the goals, where are we now? What did we achieve? What challenges we have? How are we going to approach them? What extra help we need? And also, even within the funding process has been even simplified. So our grant is within a year because this is determined by the length of the experiment. And the funding is going to be each quarter, and it could not be a much easier process. So I think, if I really want to summarize it, if you have a project that really fits the purpose of what the FFB trying to do, find the cures for people with retinal diseases, what the TRAB program is trying to do, accelerate your early data, your early preclinical in nearer to the clinics and have a partner, then I think, you should consider submitting this application. And if you are absolutely successful in submitting it, you will have a partner in developing your product. And throughout, you'll have somebody who will basically answer your question and take you through. And I applied last year for 2 NIH grants and applied for the FFB. And in comparison, the FFB is a pleasure to do. And with that, thank you very much. And I'll take any questions.
All right. Thank you, Marty, so much for your remarks. This is the Q&A section. I think we already have 2 questions primed, but I just want to let the audience know, please either direct your questions towards me or Marty. We're more than happy to answer them for you. So I believe the first question we have, if we subcontract with an institution, will FFB allow us to include the research institution's indirects in our budget? Unfortunately not. It's sort of one of our major policies is when it comes to our funding mechanisms that we do not support indirect cost. The next question is, for the TRAP, essentially the viral vector ocular inflammation PPA, should the 3+ projects address the same question, address different question, but thematically linked, or can address different aspects completely? I think it's the latter 2 is what we're looking for, is that either sort of a themed research approach where each one's bringing a different skill set to the table in order to answer a central question, or ones that are addressing maybe different aspects of at least one of the research priority areas listed for this particular program project award. I think it just, what you have to consider is the budget, I think, at that point in time. What makes the most sense for the money that is able to get used? So it seemed that 3 different aspects may be able to just scratch the surface of the research, but I think a thematically linked approach in order to answer or address one of the research priority areas is probably more so what we're looking at in order to actually get some type of solution for viral vector induced ocular inflammation. So Maya, maybe I'll ask you one question, maybe just sort of tease things out, because like I said to you earlier, I'm usually on the opposite side of it. What part, when you're developing the budget and timeline, and I'll say this, and I don't think I've told you this before, when we've had other people review your particular budget and timeline, it had a much more industry feel to it. Can you please explain to maybe the audience your process for developing that? Because it's very important to really have, I mean, we're essentially asking for people to submit a Gantt chart when it comes to product development. So for those who maybe haven't done a Gantt chart or aren't so familiar, can you please explain, maybe just give me the high level view of your process?
I think, for us is maybe different to everybody else because we are at a late preclinical stage, and we know what we need to do to be able to file an IND. And this is, FDA regulation very clearly. There's a list and a set of experiment. We have to show the molecule is safe. We have to show its kinetics. So this is I would say a head start for us. We don't have to create that. I think if you are a step behind, again, I think there is a lot of help within the FDA, within a lot of other organizations. If you are at an early product selection, if you are between candidates, if you are trying, I think, the FDA guidance for drug development is very clear. And my recommendation is look what is needed to take it to the next stage. Okay. And I think, from a drug development point of view, the success rate is really small because we talk about 5% will end up there. So I think the focus would be what I must do now to find out in the next experiments or two that I should not continue to develop it, rather continue to develop it. And I think, that is basically the philosophy, because if it's not safe, if it's, for example, oral drug but cannot be taken by mouth, it cannot be absorbed, or it could be given every 3 hours. So you have to find out all the essential bits. And I think the ultimate thing for us, which we do, is really what we call the target product profile. If your development all successful, and this is available in the clinic, what would it be? Who would use it? How often you use it? How will it work? For whom it will work? And what safety you are acceptable to those people at those circumstances. And that's usually, we are lucky because we are the late stage. We had that target in front of us. We knew the next step. But if you haven't got that, I think you need to clarify, where am I now? What is my next critical goal that I need to get to? And how am I going to get there? And I think, part of the application is a whole emphasis in the execution plan. In terms of budget, how do we work out the budget? Again, we both, how much time will it take us to do that? Because we know the activity. Who contractor we'd like to use? Why would you use them? And we approached the number of contractors and we got quotes from them. So we had a good idea when we subcontract this work or this experiment, how much they will cost. We knew our own time. We knew whether we need to use computer. I think the application prompt you to all these things. But as you said, Chad, I think the main thing for us, we did not realize at the beginning that indirect cost not included. That is, it's a fixed amount. That is given to one person, the grant. And if the person have subcontractors, it's up to them to deal with that. So from the Ffb point of view, you as an applicant, you need to work out everybody involved in your research, their cost, equipment cost, travel cost, whatever, all these elements of cost. And the Ffb will have an overall cost from you as an API.
All right, thank you for that. Appreciate it. And you mentioned some very sort of good jewels there about one thing that I did not mention during it is sort of like what happens after you're awarded. And one thing you brought up is the development of a TPP. Yeah. And just to let the audience know, that is sort of one of the things we push all awardees to do at the very beginning of the project is to develop a target product profile for your particular technology, so that you have some sort of these goals in mind, or you have the clinic in mind as you think about development. So Madi, thank you for mentioning that. Yeah. The next question we have is, are collaborators, subcontractors allowed to participate in multiple applications led by different project leaders? And the simple answer is yes. We don't have any policies that say that inhibit you. I mean, I think we look at each individual application separately from one another. However, you are sort of bringing up a good point in that for this next application call, we will only be looking to fund anywhere, depending on the level of funding, one TRAP PPA and probably 5 to 7 TRAP awards. And so those are the categories TRAP 1, 2, or 3. And the reason that I'm saying 5 to 7 is if it depends on the mix of the TRAP awards that actually get scored the best. But they're all scored together, and then sort of the funding shakes out from there. The next question is, if we have a pathway that we believe is defective in a specific RP model, but we do not yet have a therapy, will FFB fund the investigation of therapies to restore that pathway to normal? In that particular scenario, in what you described, that particular approach would not work for the TRAP award. However, if you go to the foundation's website, we have other types of awards, such as Individual Investigators Award, that that this type of scenario may fit better within. However, if you have something in hand, and for this particular case, let's just say you had a small molecule that you think could mitigate this, and you've had one experiment done to show that it has some particular efficacy on that particular pathway that you described, that would make it eligible for the TRAP award, because you have something in order for the program to advance. It will not review well if you say your main plan is you have a target pathway, and the first experiment you're going to do is a drug screening program to see if we can actually target that pathway. Because if it fails and you don't find any targets, then the whole project fails. We actually want something that we can advance. And I think actually that dovetails a little bit into the next question, which is, what is the process if the project fails and is canceled during the time frame of the grant? I think this is a great question. I will say this, that in how we approach this, I think we give the project as much flexibility and chances we can in order for it to succeed. For example, if you have a particular milestone and you say you're going to meet it this way and it just doesn't seem to work, what we'll want to know is, and we'll know that through our monthly updates that we have, is that, we're not seeing what we should. And hopefully through your project mentor, as well as the advisory council, we can figure out a way to pivot the project in order to, let's say, rescue it and actually find something that does work. So we're not just going to give up if you can't reach the milestone on your initial attempt. We will try to work together in order to find something else that works in order to mitigate that. I'll mention that the TRAP program is really made to be a flexible program. Therefore, let's say halfway through your project, a new technology comes along that's going to help your project out. We want to see that put in there. If it's going to help it be a better therapy, we'll work to adjust the research plan in order to fit that in. So it's not meant to be static. It's meant to be a living project in which we can ebb and flow as we need to in order to make it work. But if all else fails, yes, if you're halfway, let's say, done with the project, it's just not working, can't get it to work, can't reach your milestones, what we'll do is then find out basically a closeout process, sort of a soft landing for everybody. For example, we don't want you instantly firing postdocs just because of this funding, but what information can we garner, through the closeout process to inform maybe future therapy development? So we'll come up with a plan and how to close out the project and then terminate it. And that's that sort of process. But to be honest with you, I think that through our vetting and we won't come to that point. I think we'll be able to find a way to pivot the projects in order to get valuable data out of each one. That's my hope.
I think, Chad, this is what I have seen, and this is what I also just want to say about the go, no-go decisions, because when you do your experiment, I think you have to be clear. If a certain outcome comes or it doesn't come, what's your reaction? You're going to go to the next experiment, you're going to modify it, you're going to stop. Because go, no-go is really important. It doesn't mean the program is killed. It just means you're not continuing to go on your original path when it's not going to be as productive. But every step, you learn something, you modify, and you go forward.
We have a question here, is how many applications will you accept for review following the letters of intent? We don't have a particular number that we will invite for full applications. If we will invite those that we feel that have a chance to review well, that meet the criteria that are laid out in the sort of application documents. And so for example, if we get 50 applications and 40 of them meet the criteria, then we will review 40 of them. However, if we do not feel, we feel if 10 only meet the criteria that we have set forth, then 10 will be reviewed. We don't have a set number for the amount of people we will invite for a full application. Okay, I see something in the chat. Hold on. It says here we have, would TRAP support studies to repurpose an existing drug that has gone through clinical trials for another disease type? 100% yes. We already have, if you go, Dr. Paul Yang, who we have funded, is doing just that. He is looking at an approved FDA drug for an inherited retinal disease purpose for neuroprotection. So yes, that will be considered. Okay, well, let me see. I think we've answered the one question in the Q&A right now. Does anybody else have any more questions? Or Madi, do you have, we'll give it a minute. Maybe maybe I'll ask you one more question to give people time to think. Is there anything, and I'm sort of putting myself out there, is there, and don't take budget to account, I guess at this point, it's just so notional. Is there any other support that you would like to see the foundation give you? And just in any way, besides like sending more maybe postdocs or of course more money, I think would help, but we're budget limited. But as far as from a program management or programmatic standpoint, is there anything else that you think we could do better on?
I think, you know, the issue, we're all dealing with rare diseases. And a lot of these diseases, we really don't understand the natural history. And I'm thinking towards the end, it's easy when you do it in the lab. But when you go to the clinic, you need to find the right patient in the right locations. And really, I know you have the tracker. I know you have, I think, something to do with where are these patients? How can we access them? How can we approach them? I think that's very important because, we talk about, even just a simple example, we all develop drugs to cure a disease, but we have an idea about what the risk benefit is. Would I take a drug which is cause me dizzy, every time I take it for half an hour? Probably I wouldn't. But we don't know what the patient would accept. And in my experience, I'm surprised. We really do not dig into patient insights and what they need and what can they help, how can they help us to push this drug forward.
Okay. Yeah, and I think that our clinical consortium team, as far as from the Ffb being led by Todd Durham, is working diligently to try to solve that problem, and to identify. Your statement right there is, I think, one of the constant issues that we're dealing with. But I think we're working hard to find creative ways as these therapies come online to actually understand how they're going to work in the clinic, understand who is will benefit from them the most, or even a little bit at that point, right? Even if a patient will even a little bit benefit from a therapy, where they are and how they can take advantage. So yeah, I agree 100%. And what did you want to say?
Maybe just another thing, because, I think there's a lot of people trying to work on the same pathway or the same drug or repurposing a drug. And I wonder whether the Ffb, not during COVID, maybe, a few years ahead, whether we can consider pulling a group who works on the same pathway or the same drug or the same patient population together, facilitated by, by the Ffb, to really, one, we don't reinvent the wheel, but two, we learn from each other, and three, maybe prioritize the program. Maybe some of these therapy can be used in combination. Maybe, you talked earlier on about the AVV and the inflammation. There's a lot of people who work on anti-inflammatory pathways in the eyes, people working on gene therapy. So maybe putting this group together will come up with something. That's another thing I would like that to be.
Right, and I will point out that a similar workshop, like you just mentioned, like we did for the inflammation, I think that we will continue along those lines and those types of practice, and having these deep dive focus groups on particular
and I think
topics that will bring people together that are focusing on similar ideas.
Yeah, and a third area, it is not, we're going to carry the experiment. I think the ultimate aim is to bring this to the clinic or to take it through clinical trials. And I don't know the answer. I don't know how much. linkage, relationship in the background between the FDA and the FFB. Because, you know, with FFB acts as a conduit and discussion, because we know through the FDA there is formal ways of approaching them, of asking a question. It would be nice if the FFB can have an extra arm, you know, accessing the FDA or even EMA in Europe, whatever, to really get an insight for this program as they get nearer the IND submission.
Yeah, agreed. We do have another question up here. So let me, we have two more questions here. And the first one is, does FFB get any IP rights over the funded work via the TRAP grant? The current state of our policy when it comes to the TRAP grant is that no, we do not get any IP rights over the funded work. As it stands though, we do hold a first right of refusal for licensing. And the reason being is, since we're working in the rare disease space, we are very sort of, we would like an opportunity to review the technology from our RD fund side for either potential investment or to make sure that the technology is going to within an organization or a group that has the knowledge and background in order to advance that technology. The last thing we want to see is that we make these investments. They go on to get third-party investment, but then go somewhere where the technology sits on the shelf for 5 years. We don't want that to happen. We want to be able to wrap the support around these technologies in order for them to continue to develop and advance. Second question is, could you provide further details on what the monthly updates involve? Sure. The monthly updates go along a template. I will not be showing that here today, but essentially what you're doing is providing a data update to the foundation, the project mentor, and maybe an innovation advisory council member. You're also giving information on the technology transfer status, as well as highlighting any issues that you're currently having. So we track the Gantt chart as far as your development schedule, and if you're off on that, you know, we will discuss what we call get well plans for the technology. So what we're trying to do is actually in the program is, while it is research based, is bring a development flair to it, so that we're actually taking more of an engineering and sort of product development approach to the award. And we want to make sure that during the monthly meetings that if there are any problems that we're able to mitigate them immediately. The last thing we want to do is learn about a problem 6 months late, and then now we're trying to pivot and provide advice and counsel when it could have been done 5 months ago. So that's what's really involved in monthly updates. Right now, the monthly updates are lasting anywhere from 30 minutes to 1 hour. We try not to make it a time suck for you all. The template, the reporting template that we use, once populated, the only thing really that you're adding to it are pieces of data. So there's a little upfront work in order to get the template established, but after that, it's really just a storyboard for telling us about the new data that's being presented or any problems that may be occurring. All right, I will just give it a little bit of time. We are almost at time for this meeting. Are there any additional questions for myself or Dr. Farhan here? Okay, I see none. Again, first and foremost, Mahdi, thank you so much for joining us today for this Proposers Day. I hope that the audience has learned something about our application process, and I hope all of you apply. Please, please apply. We want the greatest of ideas. If you have any questions, please, you can email me at cjackson@fightingblindness.org. Please go to the website and download the documents and review them, and please apply. So with that, I thank you. This is being recorded. Go to the Foundation's YouTube channel to see it at a later time. We will try to have it up by the end of this week. And I will wish everyone a good day and take care.
Thank you. Thank you, everyone.